Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecules. So in the last lecture, we started carbohydrate and we saw the classifications of carbohydrate on the basis of different criteria. Among those, one of the most important classification was on the basis of the number of sugar molecules present in a carbohydrate. And on the basis of that, it was divided into 4 categories. That was monosaccharide, disaccharide, oligosaccharides, and polysaccharides. Then we briefly started with the structure. So in today's lecture, we will discuss the structure of these carbohydrates in detail. So what we saw in the last class is that the carbohydrates or the sugars have 2 types of structures, right? So the carbohydrates have 2 types of structures. 2 types of structures. Now, what are these 2 types? The first one is open chain. The first type is open chain, and the example you can see here. So it is a linear chain structure. And the second type is the ring structure. The second type is ring structure. Now, in the ring structure also, it has 2 types based on the type of ring. So we are seeing here 2 different types of ring, right? Okay. So first, let's see open chain. So what do we have in the open chain? The example that we are seeing here in the open chain is the example of glucose. So it has 6 carbon molecules, as you can see. Its general formula will be C6H12O6. Now, when this open chain structure, when the carbon that has the functional group, it forms a bond with any other carbon, it gets converted into the ring form. Okay. So for example, here in the case of glucose, the C1 carbon, it attacks the C5 carbon, and the ring kind of structure is formed that we are seeing here. Okay. Now, in the ring structure, this first type of the structure, if we see, we can see the structure looks something like this, right? And this structure or this ring is known as pyran ring. And so this structure is known as pyranose, and it is derived from pyran ring. Okay, it is derived from pyran ring. Now, how many carbon molecules are present in the ring? So we can see we have 1, 2, 3, 4, and 5. So the pyran ring is what? It is 5 carbon ring. It is 5 carbon ring, right? So this is one type of ring structure. Now, the second type of ring structure is that we are seeing here. So like it was the example of glucose. The second is, it is the example of fructose. So the fructose is also 6 carbon molecule, right? It is 6 carbon molecule, but the structure, if you see, it has a different structure than that of this glucose. So what is the difference here? So the difference here we can see if we start numbering. So the numbering here will start from here 1, 2, 3, 4, 5, 6. But inside the ring, you have only 4 carbon molecules, right? So here, this ring is known as, or this structure is known as furanose. This structure is known as furanose, and it is derived from furan ring. Okay, it is derived from furan ring. And this ring is, if you see, look into the structure, so the ring contains only 4 carbons, 1, 2, 3, and 4. So it is 4 carbon ring. Okay. So the ring structures are of 2 types based on the type of ring it consists. So this is about the structure. Now let's discuss about the monosaccharide structure. So in the last lecture we saw, so monosaccharides are one that has only 1 sugar molecule. Only 1 sugar molecule. Right. It has only 1 sugar molecule. But before moving to the monosaccharide structures, let's see one term that is known as carbonyl carbon, because in the upcoming slides we'll come across this term a lot. So what is carbonyl carbon? So if we look at the structure here, so what we are seeing, if we look at the structure, we are seeing that there is one carbon to which the functional group is attached, right? This is the example of glucose, right? Similarly, there is one more structure, open chain structure, that is fructose. Here what we are seeing, that here also there is one functional group, CO. So the monosaccharides or the carbohydrates consist of two types of functional group, either the aldehyde, that is CHO, or the CO group, right? Now, what is a carbonyl carbon? So this is the carbonyl carbon actually. So carbonyl carbon is the what? It is a carbon on which carbonyl group is attached. Okay. So the carbonyl carbon is the carbon on which carbonyl group is attached. So in the linear structure, it is very much clear that where the carbonyl group is attached. But what about the ring structure? So if we take the example of glucose, what we are seeing that this CO, this CHO group, it forms bond to CHO, this fifth carbon, right? And if we start numbering, so this is your sixth carbon, right? Because this CS2OH, it is clearly visible in the ring structure as well. This is your CS2OH, right? This is your CS2OH. So this is sixth carbon. This will be your fifth carbon. This will be your fourth. This will be third, second, and first. So what we are seeing that in the case of glucose or in the case of pyranose structures, the carbonyl carbon is here. This is your carbonyl carbon, right? So in the case of glucose, the carbonyl carbon is C1 carbon. In glucose, carbonyl carbon. In glucose, carbonyl carbon is carbon number 1, or I'm representing it by C1, right? Now, what about fructose? So in the case of fructose, if you see, this is your open chain structure. So this is 1, this is 2, this is 3, 4, 5, and 6. Now, here what happens? That this CO, it again attacks the fifth carbon, right? And form the ring structure. But in this case, the numbering will start because it has CS2OH first. So numbering will start from here. This is 1, this is 2, this is 3, this is 4, this is 5, and this is 6, right? So in this carbon, in this case, which is the carbonyl carbon? This one, or carbon second. So in fructose, carbonyl carbon will be, or carbonyl carbon is C2. Okay. So in the case of glucose, the carbonyl carbon is C1, while in the case of fructose, the carbonyl carbon is C2. Now, with this background in mind, now let's see certain other terms. So the first term is anomers. So what are anomers? Before that, let's see these structures. Okay. So the first structure here that I have drawn is the structure of glucose. It is mentioned. So it is a ring structure of glucose. Okay. Now, the second structure, so this is your first structure, right? And it has the numbering starting with 1, 2, 3, 4, 5, and 6. Now, this is your second structure, right? In the second structure, if you see, what is the difference between the first and the second structure? The second structure also is of glucose. Okay. But there is certain difference. So if we look properly, the difference is in this reason. The difference is here, and in this case, here. Right. This is your H. So the difference what you are seeing is here. What kind of difference we are seeing? That in one case, the OH is located downward, and in the second case, it is arranged upward, right? It is arranged upward. So, and it is said that then what is the name of these two structures? So the first one, both of these are glucose, but their naming is different. So in the first case, where the OH is below, this is known as alpha glucose. Okay. So this is known as alpha and D glucose. D because I have told in the previous class also that D is because the glucose or the most of the naturally occurring carbohydrates are dextrorotatory. That's why we are writing D. But based on the orientation of OH, it is downward. hence it is known as alpha D glucose. And in the second case, the OH is upward, hence it is known as beta D glucose. Okay. And this, it is said that this alpha D glucose and beta D glucose are anomers. Okay. Now, next, what do we have? We have this structure of fructose, right? We have just seen in the previous slide. This is also fructose. This is also fructose. But it has difference at one position that is clearly mentioned. So the position is this one. It has difference at this position. And what we are seeing, in one case, the OH is located or is oriented downside. And this is known as alpha D fructose. And in the second, since it is upward, it is known as beta D fructose. And it is also said that alpha D fructose and beta D fructose are anomers. Now, the question is, what are anomers? So, what are anomers? So, what we can see from this, or what we can say from this thing, that there is change in the orientation of hydroxyl group, right? And this change, so anomers you can write that difference in the, so these are isomers because they have same chemical formula. So anomers are what? These are isomers that have change in the orientation, of hydroxyl group. OH group is hydroxyl group at one carbon position. And what is that carbon position? So if you look at the structure, this change is not random. It is present at a particular carbon position. And what is this carbon position? This is the carbonyl carbon, right? From the previous slide, if you see, these are the carbonyl carbon. So anomers are the what? These are the isomers that have changed in the orientation of OH group at carbonyl carbon. Okay, so these are anomers. Now, in case of glucose, the anomers alpha D glucose and beta D glucose are anomers at. So alpha D glucose and beta D glucose are anomers at carbon 1 position. Why? Because in the case of glucose, carbon 1 is the carbonyl carbon. While in the case of fructose, these are anomers. Alpha D fructose and beta D fructose are anomers at carbon 2 position, because in the case of fructose, second carbon is the carbonyl carbon. Okay, so this is about anomers. Now, the next thing that we'll see is epimers. Okay, again, what are epimers? We'll see with example. So the structure that you are seeing here is what? This structure. So it is 6 carbon ring, right? It has 1, 2, 3, 4, 5, and 6 carbon. And it has OH on the top, or have orientation above the plane. So this OH, this structure will be called as beta D glucose, right? This is called as beta D glucose. Now, let me draw one structure. So there's one structure that looks something like this. Okay, this structure looks something like this. It has OH, H, OH, H, similar to what I have just drawn. OH, H, and OH, H. Now, if you look at the structure, we have the numbering 1, 2, 3, 4, 5, and 6. If you look at the structure of glucose and this structure, what difference can we observe? So the difference that we can observe by looking at this structure is at one point, right? So the structure difference is at this position. In this case, the orientation, and in this case, what is the orientation? So this is the fourth position, right? And this structure is known as beta D galactose. This is known as beta D galactose. Now, it is said that beta D glucose, or you can simply write glucose and galactose are epimers. Are epimers. Okay. Now, let me draw another structure. Let me draw another structure. So this one is beta degalactose. Now, you have one more structure, something like this. Okay. So you have CS2OH, HOH, HOH, OH, H, and OH. Now, if you look at this structure and compare this structure with the beta D-glucose structure, at what position we can observe the difference? So the position at which we can observe the difference, here we can number 1, 2, 3, 4, 5, and 6. So the difference will be, let me use different pen. So what difference we can observe? So the difference will be, if we compare this structure, difference will be here and here, right? In this case, the OH is above the plane, and while in the glucose case, it is down. And this structure is known as beta D mannose. This structure is known as beta D mannose. Okay, and it has difference at a certain carbon position, and it is also known as that glucose and mannose are epimers. Okay, are epimers. So by this, if someone asks that what is epimer, so by looking at this, what we can write. So what are epimers? We are seeing that all these three structures have same chemical formula, right? What is difference? There is difference in their structure. So epimers are, again, if they have same chemical formula and different structure, this will be isomers, right? So epimers are also isomers in which there is change in the orientation of hydroxyl group, right? Hydroxyl group, where at any carbon, right? At any carbon, any one, not okay, at any one carbon, but apart from or except carbonyl carbon, right? Except carbonyl carbon. Why carbonyl carbon? Because if the change will be at the carbonyl carbon, that will be known as anomer, right? So what are epimers? Epimers are basically the isomers in which there is change in the orientation of hydroxyl group at any one carbon except the carbonyl carbon. Okay. For example, in the case of glucose and galactose, these are epimers. Why? Because they have changed at the position of carbon 4. So glucose and galactose are epimers at carbon 4th position, right? At carbon 4th position. While the glucose and mannose are epimers at carbon of 2nd position, right? At 2nd position of the carbon, right? So this is about the epimers. Now the question is galactose and mannose also epimer? So if you look at the structure, so what differences can we observe? We can observe one difference here, right? At the 2nd position. At the 2nd position, there is a change in the orientation of galactose and mannose, while the second difference will be at position 4th. That is, in this galactose, the OH is upward, while in mannose, the OH is downward. But are these epimers? The answer is no. Why? Because in the case of this, what we have studied about the definition of epimer, that the change should be at one carbon. And in this case, the change is at 2 carbon, right? So we can write galactose and mannose are not epimers, because in this case, the changes are at 2 positions. Okay. So this is about epimer. And along with this, we have also seen the structures of galactose and mannose. Those are also These are also 6 carbon sugar molecules, right? Now, next thing that we'll see. So till now, we saw the structure of glucose, galactose, and mannose. and these three were 6 carbon sugars. Now let's see the structure of a 5 carbon sugar. So the 5 carbon sugar, the one of the example is ribose. So this is the example of ribose. Ribose is a 5 carbon sugar. Okay, so this will be your H, this will be your H, this will be your H. And if start numbering, 1, 2, 3, 4, and this is a 5th carbon. So it is 5 carbon sugar. And if you look at this linear structure, how it will be? So the linear structure will be like this. 1, 2, 3, 4, 5. CS2OH, right? So this is the linear structure of ribose. Now, in the case of ribose, where is the carbonyl carbon? This one. So this is your carbonyl carbon. This is the carbonyl carbon. First carbon is a carbonyl carbon. Okay. Now, with the help of this ribose, our body can derive one more sugar, and this sugar has a structure something like this. So the ribose, the formula will be C5H10O5, right? Now, with the help of ribose, one more sugar can be produced, and this sugar will look something like this. This will look something like this. Okay. This will look something like this. It has OH, H, H, and H. So the difference is at carbon second, right? At carbon second, instead of this OH, it has only H. So it do not have this oxygen molecule, and because of this, it is known as deoxyribose. Deoxyribose is present in a DNA. We will see this when we will study about nucleic acid. So for now, this is the structure of deoxyribose, and it do not have hydroxyl at second position of the this ring. Okay, and the deoxyribose is derived from ribose, and it is also known as derived sugar. It is known as derived sugar. And what will be the formula of deoxyribose? The formula will be C5H10, and since it does not have one oxygen molecule, it will become O4. So this is about the structure of monosaccharides. Now, just to remember, I have summarized all the structures here. So the first two structures that we are seeing is what? The first one is OH is below, so it's alpha glucose. I'm skipping writing D, so it should be already understood. So it's alpha glucose, and the second structure, since OH is above, so this is beta glucose, right? Here the structure is also 6 carbon, but it has furan ring, so it is fructose, right? And since OH is downward or below, so it will be alpha fructose, and this will be OH is above, so it will be beta fructose. Beta fructose. Now, here what do we have? We have this OH. It is 6 carbon, but the OH at 4th position. The OH at 4th position is above. So this is what, and this OH at 1st position is also above. That makes it alpha galactose, right? Sorry, beta galactose. That makes it beta galactose. And if we want to draw the structure of alpha galactose, everything will be same, right? Everything will be same, except here the OH will be down, right? And this OH is up. So this will become your alpha galactose, right? Now, what is the next structure? So this structure, how many carbon molecules do we have? We have 1, 2, 3, 4, and 5. So this is 5 carbon molecule and is furan ring. So it is what? It is known as ribose. And since OH is above, this carbonyl carbon OH is above, it will become beta ribose. While in the second case, it should be like this. It should be OH and H. Okay. It should be OH and H, and hence it will become alpha ribose. So these are all the important structures of the monosaccharide that you should remember, that how these structures are made. Now, the next thing that we will study is about the reducing and non-reducing sugars. So what are reducing and non-reducing sugars? So as we know, what is term reducing means? It means that it is reducing something, or it is causing the gain of electron from somewhere, right? Now what? So this carbohydrates or the sugar molecules, they gives this test. They give this reducing. They have this ability. Some of the sugar molecules have the ability to reduce something while others don't. Okay, how we know this? So for this we use certain reagents. Okay, so the reagents to test reducing ability of sugar. So there are reagents to test the reducing ability of sugars. Okay, so mainly there are two type of uh, this reagent. So first one is known as Fehling reagent. The first one is known as Fehling reagent, and the second is known as tolerance reagent. Second is known as tolerance reagent. Now, what do we have in the Fehling reagent? So in the Fehling reagent, originally it has cupric ion, that is Cu2 plus. It is the cupric ion. Now, when a sugar or anything reduces this, so it will reduce, mean it will accept electrons, right? So the charge will decrease. So it will become cuprous ion. Okay, so in Fehling reagent, upon reduction, the cupric ion gets converted into the cuprous ion. While in the case of tolerance reagent, it consists of silver ion. Okay, and this silver ion, upon reduction, so when it's electron, upon reduction, it becomes silver, right? So these are the two reagents, and when these are reduced, they change the color or they give certain color, and on the basis of color, one say that the reagent is reducing or not. Okay, sorry, the test molecule or the sugar is uh, reducing or not. Okay, now, so what does reducing sugars do? So the sugars, reducing sugars, what they do? They reduces. Definitely, they will reduce this reagents. They reduce Fehling and tolerance reagent, which means that it will convert, this reducing sugars will convert this cupric ion and silver ion into cuprous and silver. Cuprous and silver, right? Now, why some of the sugars does this while others don't? So the sugars, they reduce this reagent because they have free carbonyl group. Because these sugars have free, they have free carbonyl carbon, sorry, not group, they are free carbonyl carbon. Now, what do I mean by free carbonyl carbon? Free carbonyl carbon means that they are not involved in any bond formation. So they are not involved in any bond formation. Okay, they are not involved in any bond formation. So if you look at these structures, now these are the major monosaccharide structures, right? So this is your carbonyl carbon. This is your carbonyl carbon, right? This is your carbonyl carbon. So if you see any of these carbonyl carbon, none of these carbonyl carbons are involved in any other bond formation, right? These are not involved in bond formation. So if we talk about the examples of reducing sugars, so the examples of reducing sugars are all monosaccharides. All monosaccharides. Why? Because none of these monosaccharides, carbonyl carbon is not free. Right? They have free carbonyl carbons because they are not involved in any kind of bond formation, and hence these are reducing sugars. Apart from monosaccharides, we will see that disaccharides are also reducing, but we have certain exceptions. Okay, so all monosaccharides are reducing. Apart from this, we have disaccharides such as lactose, okay, maltose. These are also reducing in nature. Okay, now one important point. sucrose is non-reducing. Okay, sucrose is non-reducing. So we will see the structure of sucrose, but with this discussion, we can tell that why sucrose is non-reducing, because probably its carbonyl carbon is not free, right? And that must be involved in certain bond formation. So the carbonyl carbon of sucrose is involved in bond formation. Okay, involved in bond formation. And because of this reason, the sucrose is non-reducing. So this is about the reducing and non-reducing sucrose. Okay. Now let's talk about the structure of disaccharides. So what are disaccharides? We have seen in the last class that disaccharides are what? Disaccharides are, it consists of two sugar molecules. So disaccharides consist of two sugar molecules, or when you hydrolyze disaccharides, two different or two same kind of sugar molecules can be formed. Okay, it consists of two sugar molecules, and how they are linked. So in the case of carbohydrate, the linkage is linked by glycosidic bonds. We saw the formation of glycosidic bonds. So they are linked by glycosidic bonds. And how glycosidic bonds are formed? So glycosidic bonds, how they are formed? Formed by release of water molecule, right? They are formed by release of water molecules, right? So suppose this is your one sugar molecule, this is your second sugar molecule. So the water molecule will come from one molecule OH and the second molecule H, right? So this will lead to these water molecule will be released, and you will have one structure. You will have a structure something like this, right? You will have a structure something like this. Okay. So this will be the structure, and this will be your glycosidic bond. Okay. Now, if we talk about the types of the glycosidic bond, we have two types. The first type is known as alpha bond, alpha linkage, let's say, or alpha glycosidic bond. So what do we have in alpha linkage? So the alpha linkage will look something like this, the structure that I have drawn. So this is alpha linkage. So from where it will be formed? So suppose your sugar molecules, both of the sugar molecules are alpha. Okay. So this is, say, alpha glucose. This is alpha glucose. Okay. Because it will have CH2OH. And again, you have another alpha glucose. Okay. You have another alpha glucose. So OH is here. This is your another OH. This is your CH2OH. So this is again alpha glucose. So in this case, when the bond formation will take place, it will look something like this. Here, the OH of this OH and this H, it will together makes the water molecule and it will release, and the bond will look something like this. And this bond will be called as alpha, because this is 1, 2, 3, 4, 5, and 6. And again here 1, 2, 3, 4, 5, and 6. So what we are seeing, that the first carbon of one glucose and the fourth carbon of second glucose is making the bond. So the bond that will represent is, because it is linear, it will be alpha and then 1, 4. Okay, because first carbon of the one glucose molecule and fourth carbon of another molecule is making the bond. So it will form alpha 1,4 glycosidic linkage. It will form alpha 1,4 glycosidic linkage. Okay. So this is one type of linkage. The second, what do we have? We have beta linkage. Now, what do we have in the beta linkage? So in beta linkage, suppose this is any sugar molecule and it is looking something like this. It is looking OH, this is H. You have another sugar molecule and it is looking something like this. It has OH molecule at the top. Okay. and this is H. So in this case, this hydroxyl group and this H won't participate in bond formation, but this OH and this H will participate in bond formation, and it will cause release of water. And the bond that will be formed will look something like this. So this is your one, this is your second, right? So the bond that will be formed will be something like this. So it will be not linear like this, it will be in this shape. Okay. So this will be the bond, and this will be called as beta glycosidic linkage. And again, this is 1, 2, 3, 4. So this will also form beta 1,4 linkage, right? So it will also form beta 1,4 linkage. So this is how we name the glycosidic bonds. We will see it more in upcoming slides when we draw the structure of disaccharides. Okay. So this is disaccharide structure. So the first structure of the disaccharide that we will study is maltose. So maltose, where it is present? It is present in malt sugars. It is present in malt sugars. And what it is made up of? So the maltose is, it is made up of alpha D glucose, and alpha D glucose, like 2 alpha D glucose together makes maltose. So alpha D glucose and alpha D glucose. So the structure that we are seeing here is alpha D glucose, and second is also alpha D glucose. Okay. Now, when the when the bond formation will take place, how will it look like? The structure will look like something like this. So structure will be something like this. It will be. So you have one. So this is OH, this is OH, OH, CS2OH. Similar will be the case here also, right? The OH, OH, right? So this is the structure of maltose. Okay. Now, if we talk about the linkage, what kind of linkage we are seeing here? So if you number, this is a 1, 2, 3, 4, 5, 6. And here also 1, 2, 3, 4, 5, 6. So if you look at the numbering, what carbon molecules are involved in bond formation and what kind of linkage it is? So the linkage we are seeing is linear, something like this, right? And this kind of linkage is what? Alpha linkage, right? And the second thing, what carbon molecules are involved? So of the first glucose, the first carbon molecule is involved, and of the second glucose, the fourth carbon molecule is involved. Hence, it becomes alpha 1,4 glycosidic linkage. So in case of maltose, the kind of linkage we have is alpha 1,4 linkage, right? Then this is the structure that I just drawn. So this is similar to what I have drawn. I have just added for the clarity purpose. Okay. Now, one more important thing to notice here is that the carbonyl group, even after the linkage, one of the carbonyl group is free, right? This carbonyl group is free, and hence this sugar becomes reducing, right? So because what are reducing sugars? The reducing sugars are the one whose carbon is free. So in the case of maltose, we can clearly see the carbon is free. Hence, since it has free carbonyl carbon, so it is what? Reducing sugar. It is reducing sugar. Okay. So this is the maltose, and it is also the example of alpha 1,4 glycosidic linkage, right? Now, the next structure that we'll study is lactose. So what is lactose? So lactose is made up of beta D galactose. It is made up of beta D galactose and beta D glucose and beta D glucose. Okay. And where this lactose is present? Lactose is present in milk sugar. It is present in milk sugar. Okay. Now, the first structure here, what it is? So we can see it by the orientation. First start with carbon 1, So here the OH is above, it means it is beta. And since the fourth OH is also above, so it is galactose, right? It is beta D galactose. Now the second structure, what do we have? We have OH above, hence it is beta. The fourth carbon OH is down. It means it is glucose. Beta D glucose, right? So what kind of bond or what kind of structure will be formed here? So the structure that will be formed, if you see, so it will be something like this. So this is your galactose. This is your galactose. And the second is something like this. This is your glucose, right? This is your beta D glucose. Now the bond, how the bond formation will take place? The bond will form something like this, right? So this bond is like this. Okay, this is the glycosidic bond, and this is the first position here. 1, 2, 3, 4th, 5th, right? So this is their fourth position. But this, since the bond is like this, so it will be beta 1, 4 glycosidic linkage. It is beta 1, 4 glycosidic linkage. And this is the structure. This is what I was talking about. So this is the bond. This is the kind of bond that is formed, right? And hence it is beta 1, 4 glycosidic bond, because this is carbon number 1, this is carbon number 4. Again, if you look at the carbonyl carbon, in this case also carbonyl carbon is free. So since the carbonyl carbon is free, lactose is reducing, right? Since it is free, lactose is reducing. Okay, so lactose is also reducing. Now the third type of disaccharide that we will study is sucrose. Okay, now this sucrose is made up of it is made up of what? Alpha D glucose. It is made up of alpha D glucose and beta D fructose. Okay, it is made up of alpha D glucose and beta D fructose. Okay, and this sucrose, it is like the table sugar. So the sugar that we use, right? Table sugar, sugar cane. So the sucrose are present in table sugar, sugar cane. Now let's draw the structure of sucrose. So what it is saying, it has alpha D glucose. Okay, it has alpha D glucose. So how does the structure of alpha D glucose looks like? Alpha है, so it will have OH below. This is your CS2OH. I'm just drawing this much. The rest of the diagram we know. So this is alpha D glucose. Now, what else we have? We have beta D fructose. So what is the structure of beta D fructose? So the beta D fructose will look something like this. Okay, sorry, the beta D fructose will look something like this. So since it is beta, the OH will be above, right? And this will be your CS2OH. This will be your CS2OH and H. Okay, and rest. Okay. So this is first position. Similarly, this is first position. Sorry, this is first position. This is second. This is second position. But in the case of glucose, so this is alpha D glucose, and this is beta D fructose. Now, in sucrose, what happens? In sucrose, the bond formation will take place between this first and second carbon. Okay, so OH of first carbons and H of the second carbon of fructose, they will involve in the bond formation, and it will cause release of water molecule. It will cause release of water molecule, and hence the structure that will be formed, will look something like this. The structure will look something like this. Okay, so it will look, we have, this is your fructose, and this is your second position. This is your CS2OH. This is your CS2H. Just a second. Yeah, so this is your CS2OH. Fourth, first, second, third, fourth, fifth, and sixth. So the bond will look something like this. Okay. So and this bond is known as this linkage is known as beta 1 2, sorry, because it has glucose and fructose both, so its bond will be something different. Okay, so its name will be something different. So the name will be the first carbon, the first moiety that we have is glucose, right? So in the first carbon we have glucose, so we'll write blue. Okay, now its first carbon is involved in bond formation, so it will be 1 alpha because it's alpha glucose, right? So it will be 1 alpha. And in the second carbon, we have beta fructose, right? And its second carbon is involved in the bond formation, so it will become 2 beta. And since it is fructose structure, we can write fructose FRU, and this is glucose 1 alpha 2 beta fructose glycosidic linkage. Glycosidic linkage. Okay. So this is how the name works, because we have alpha glucose, and of this glucose, first carbon molecule is involved in the bond formation, and we have beta fructose, in which the second carbon molecule is involved in the bond formation. So the name will be glucose 1 alpha 2 beta fructose glycosidic linkage, and this kind of linkage is present in the sucrose. Now, what is the important thing? So in the case of sucrose, we studied that it is non-reducing in nature, right? We saw that sucrose is non-reducing. Now we can see through the structure that what is the reason, right? Because here what we have seen, this the first structure is of glucose, and its carbonyl carbon is this one. In the case of fructose, the carbonyl carbon is second carbon, right? And both of these are involved in the bond formation. So the carbonyl carbon is not free, and because of this reason, sucrose is non-reducing. Right? Okay. Now, apart from non-reducing, sucrose is also known as invert sugar. It is also known as invert sugar. Now, what is invert sugar? So, the sucrose, they are originally dextrorotatory. Okay. They are dextrorotatory. Now, what do you mean by dextrorotatory? It means that sucrose, they rotate plane polarized light. They'll rotate plane polarized light in right direction, in right or clockwise direction. Okay, so when you pass the plane polarized light through this sucrose mixture, it will rotate it into the clockwise direction, right? But what happens if you hydrolyze sucrose? Okay, so upon hydrolysis of sucrose, so suppose this is your sucrose, you have sucrose mixture. Okay, you have sucrose mixture. Now you hydrolyze it. So you do the hydrolysis. So if you do the hydrolysis of sucrose, what will be present in the mixture? So the sucrose is made up of what? Glucose and fructose. So the mixture will contain glucose plus fructose, and that is alpha D glucose and beta, right? Beta fructose. Okay. Now, if you pass plane polarized light, through this mixture, okay. Now the light will rotate in the left direction. Light will rotate in left direction. That makes this mixture levorotatory. So this mixture become what? Levorotatory. So because of this reason, because the sucrose originally is dextrorotatory, but if we hydrolyze the sucrose, the mixture that will be produced will become levorotatory because it will rotate the light in the left direction. Because of this, it is known as invert sugar, because it changes the direction of the sugar. Changes the direction of the plain polarized, right, upon hydrolysis. And due to this, the sucrose is known as invert sugar. Okay. So this is about the major or the most commonly used monosaccharides, disaccharides. But apart from this, let's see two more. So this is one, sucrose is one non-reducing sugar. So there are certain other non-reducing sugars. So majority of sugars are reducing in nature, but there are certain other non-reducing sugars. So the one example is sucrose. The second example is trehalose. Trehalose. Okay. So apart from sucrose, the another is trehalose. It is also a disaccharide, which is made up of glucose, like two glucose molecules. So now we can ask that the maltose was also made up of two glucose molecules, and trehalose is also made up of two glucose molecules. So what is the difference? So difference is in their linkage. So in the case of maltose, the linkage was between alpha 1,4, right? Between first and fourth molecule of carbon. While in the case of trehalose, it is alpha 1,1. So it is alpha 1,1 linkage. Okay. So if you imagine the alpha 1, like first carbon of one glucose and first carbon of another glucose. And we know that in the case of glucose, the first carbon is what? It is a carbonyl carbon. So both the carbonyl carbons are involved in bond formation. Hence, the trehalose become non-reducing. The second example of non-reducing sugar is raffinose. Now, the raffinose, it is not a disaccharide, but it is a trisaccharide. Okay, it is a trisaccharide that is made up of 3 sugar molecules, and these 3 sugar molecules are what? Galactose plus glucose plus fructose. Okay, so the 3 sugar molecules are galactose, glucose, and fructose. And due to the bond formation between them is done in such a way that none of the carbonyl group is free. The raffinose is also non-reducing in nature. Okay, so this is about the structure of disaccharides. So we have seen the structure of monosaccharides and 3 major disaccharides, that is maltose, lactose, and sucrose. And we saw that in all the 3, there are different kind of linkages or glycosidic bonds. Next, we'll study about the polysaccharide structures. So the polysaccharides, we have also discussed in previous class that the polysaccharides, these are of 2 types or based on their function. So based on function, polysaccharides are of 2 types. Polysaccharides are of 2 types. The first one is storage polysaccharide. First one is storage polysaccharide. Now, what are storage polysaccharides? The examples are starch, glycogen, and inulin. Okay. And the second is structural polysaccharides. Second is structural polysaccharides. Okay. And what are the example of structural polysaccharides? There are multiple, but major are cellulose, chitin, and peptidoglycan. Okay. Now we will see the structure and study about these polysaccharides one by one. So let's start with the storage polysaccharide. And the first one is the starch. So what is starch? So starch, it is the polymer of alpha D glucose. Okay. It is a polymer of alpha D glucose. Okay. And since it consists of only one type of monomer molecules, that is alpha D glucose, starch is homopolymer. It is homopolymer in nature. And structure, if you'll see, it is branched. Okay. Now, how does the starch looks like? So starch looks something like this. Okay. So this is just a 2 chains, but it has multiple chains and multiple branches. Okay. So what we are seeing that in this structure, it is written, it has two type of glycosidic linkage or glucoside linkage. The one is the main one or the majorly present is alpha 1 4. Okay. So this linkage, if you see this linkage, this, what we are seeing that these are alpha glucose and the linkage is between 1 and 4 molecule, fourth and first molecule. Hence it is alpha 1 4 linkage, right? So this is alpha 1 4 linkage. But apart from alpha 1 4 linkage, we can also see a different type of linkages as well, right? There's different type of linkage as well. And this linkage is present here, right? This is present here. And this linkage is known as alpha 1 6 linkage. Why? Because if you see, this linkage is between first and sixth carbon. First carbon of one sugar molecule and sixth carbon of another. Hence, it is known as alpha 1 6 glucoside linkage or glycosidic linkage. Okay. So the starch have two linkages, two type of linkages. So we can write the starch have two glycosidic bonds, two types, not two types of glycosidic bonds. Okay. And secondly, now this starch, they are digested by enzyme. Which enzyme? They are digested. The starch is digested by enzyme known as amylase. Digested by amylase enzymes. Okay. Now, what will we find upon hydrolysis of starch? So, upon hydrolysis, if you hydrolyze starch, what do we find? So, it depends what kind of bond is breaking. Okay. So, upon hydrolysis, if break 1 4 bond, like 1 4 glycosidic bond, then what you will get is maltose. You will get maltose. Okay. And if you will break 1 6 bond, then you will get isomaltose. So in the first type or first round of hydrolysis, you will get this much structure together. You will get this much structure together. Okay. When you will further hydrolyze it, you will get glucose. That's true. But if you hydrolyze first time, you will get structure something like this. Okay. Now, what else do we have to study about the starch? So if you look at the starch, so this is only two lines, but if you draw the starch, it will look something like this. Okay. It will look something like this. Right? It will look something like this. Now, in this, if you see, it has two parts. The first part that is completely linear, right? And the second part that is has some kind of branching. So this is the first part, this is the second part. Now, the first reason is known as amylose. The first part is known as amylose, and it is what? It is linear polysaccharide. It is linear, right? It is linear. Okay. So it is linear, and it constitutes 20% of the starch. It constitutes 20% of the starch. And this is water soluble. This is water soluble, and it has alpha 1 4 linkage. Okay. Since it is linear, it has alpha 1 4 linkage, and this structure is helical. It has helical structure. Okay. So it will look something like this. Okay. Now, the branch part or the second part is amylopectin. It is known as amylopectin. And it is what? It is branched. Right? So this we can see it has branchings. This has branchings. Okay. And it constitutes 80 to 90% of the starch. 80 to 90% of the starch. It is water insoluble. And it is water insoluble. And it has both. What kind of linkage it will have? It will have both alpha 1 4 and alpha 1 6 linkage. Okay. So this is all about the starch, which is the what it is. What structural? Storage polysaccharide, right? It is storage polysaccharide. Now, in the next class, we will study about the more polysaccharide, that is glycogen, inulin, and other structural polysaccharides. Okay, that's all for today. Thank you.